Okay, this is part 2, the light reactions. So chloroplasts are like little solar-powered chemical factories. They have the thylakoids that are like the machinery that transform light energy into the chemical energy of ATP and NADPH. And so this happens inside those structures called the thylakoids. And so, again, a big picture summary is that the light reactions split water, release oxygen as a byproduct. They reduce NADP plus to NADPH and make ATP. So that's kind of the big picture. But first, before we go into the details of how that happens, let's talk about light. So light is a big, factor in this. And so light is electromagnetic energy, also called electromagnetic radiation. So this is where physics comes in a little bit. So again, if you're a bio major, you're probably going to have to also take physics at some point, either before you transfer or after. And it does make sense. It all plays part into how, the physical world works. So light is one form of energy, electromagnetic energy. And so it travels in rhythmic waves. So wavelength is a measure of the distance between peaks of electromagnetic waves. So yeah, this is a representation of the figure of waves. And so wavelength is the distance between peaks of the electromagnetic waves. So the peak is like the topmost part, and then the trough is the bottom. But a wavelength is how you go from the distance between the top of one to the top of the next one. And so wavelength can range from less than a nanometer, which are gamma rays, to more than a kilometer, which are radio waves. And I'll show a figure in a couple slides. So the electromagnetic spectrum is the entire range of electromagnetic energy or radiation. Visible light is just part of it. So visible light ranges from the wavelengths of 380 nanometers to 740 nanometers. And that visible light is what drives photosynthesis and produces the colors seen by the human eye. Really crazy to think about. So at the top is the whole electromagnetic spectrum. So going from gamma rays to the longer radio waves. And they are measured in, remember, metric units have different prefixes. So that's what helps us go from the same thing. We don't have to convert. So from meters, many meters, to 1 meter, or to even smaller units of the nanometers. We can move more easily between those different units. So anyway, visible light is just a portion of that. There's waves for all these different things, and visible light just happens to be the wavelengths that our eyes are able to see. And so visible light ranges again from 380 nanometers to 740 nanometers. And so the shorter the wavelength, actually the higher the energy. And then the longer the wavelength, the lower the energy. And I like the next figure because it actually shows the different wavelengths for the different colors. I don't remember where I found this one. So this figure is flipped. Actually, the range is flipped. So the smaller ones are over here, and then the longer ones are over here in this figure. But I like that it breaks down the colors and how they have different wavelengths. So the longer wavelengths of red, it's a longer distance between the peak of each one. Versus orange is a little bit shorter, and each one is a little bit shorter and shorter. And so this also makes a difference to our eyes, to what is visible. And again, as mentioned, the wavelengths also correspond to energy. And so shorter wavelength means higher energy, longer wavelength is a lower energy. And so light can act like that, like waves. And light is also interesting that it behaves like discrete particles called photons. And so each photon has a fixed quantity of energy that is inversely related to the wavelength of light. So shorter wavelengths actually have more energy per photon of light. And so pigments, when we talk about pigments, maybe that sounds familiar, it's any kind of substance that absorbs visible light. So there are pigments in paint molecules, right? In paints, dyes. So any kind of clothing and stuff, there are dyes, and they have pigments. So pigments are any kind of substances that absorb visible light. And depending on what they absorb and what they reflect is what color that pigment looks. So yeah, different pigments absorb different wavelengths, and the wavelengths that are absorbed disappear, and the wavelengths that are not absorbed or are reflected or transmitted. And so that's why leaves are green, is because they get light from the sun that has like all the wavelengths. And most, leaves appear green because chlorophyll is a pigment and it absorbs violet, blue, and red light, but it reflects and transmits green light. So it's kind of like absorbing everything else. And the green light is what gets reflected back or like let through, so transmitted. So this is why it's kind of two different terms. Reflected like bounces off, or it's what is allowed to like pass through. So anyway, green light is what gets reflected or transmitted, and that's why we see green. So even though the plants are absorbing everything else from the white light, it's just showing green back at us, and that's why we see green, and that's why leaves are green, because of all the chlorophyll that are in the chloroplasts and giving the leaves those colors. And so we can study this in a lab and kind of quantify light. So a spectrophotometer is a machine that measures a pigment's ability to absorb different wavelengths. I'll show a picture on the next slide. So it sends light through pigments and then measures the fraction of light that is transmitted at each wavelength. So how much light is let through at each wavelength. And you can kind of like measure out what's called an absorption spectrum. It's a way that you're shining a bunch of different lights at something and you're seeing how much gets transmitted. And so you're plotting a graph of all these different data points that plots a pigment's light absorption versus wavelength. So for every single wavelength in this range, how much gets absorbed and how much gets like transmitted. So here's how you would do that and kind of how this works. So inside of a machine, a spectrophotometer, there are these kind of pieces. So it doesn't look like this, but this is what's inside basically. There is a light bulb and then a prism. And that prism is going to break that white light into all the different colors and wavelengths. And it'll only let a certain wavelength through at a time. So it can produce all of these, but it's only going to test one wavelength at a time. And so you put in the number of wavelength for whatever like color you're trying to look at. So for green light, you would put, a certain wavelength. This allows just that wavelength through. And so say, for example, you have a solution of chlorophyll. You ground up and extracted the chlorophyll from a leaf. And so if you have that in a tube and you shine the green light through, there is a tube that is measuring how much light comes through. And so the transmittance means this is how much light is able to shine through. And if a lot is able to shine through, then that means not a lot is getting absorbed at this, by this pigment. So in this case, chlorophyll absorbs very little green light, which makes sense because it's green. So we already know as a fact, like it's not gonna absorb green. It reflects and transmits green. So yeah, it's gonna be really low absorption, high transmittance. It's letting a lot through, not a lot's getting absorbed. But if you were to change the wavelength that's getting shown, so say it's a wavelength that corresponds to like blue, and you shine this blue light through the sample, chlorophyll does absorb a lot of blue. And so that means a lot of it's going to be absorbed, not a lot is going to be transmitted through. So in this case, for this wavelength that you shine, it's going to be a low transmittance because there's a high absorption. And that means that chlorophyll absorbs most blue light. So that's how we know what we know about, what wavelengths and what colors chlorophyll absorbs best. So that's how we know, chlorophyll absorbs violet to blue and red light. And there's actually 3 kinds of pigments in chloroplasts. So there's not just one chlorophyll, there's different ones actually. So 3 types of pigments inside the chloroplasts. Chlorophyll A absorbs violet, blue, and red. Chlorophyll B absorbs blue and orange. And then carotenoids absorb blue. So you see there's a little bit of overlap, like all 3 tend to absorb blue, but then some absorb ones that others don't. And so this is kind of how you can capture the most energy. Having like a little range of what gets absorbed. So the absorption spectrum of chlorophyll A indicates that violet, blue, and red light will work best for photosynthesis. So if you ever get like a grow light, I have one like right here in my room. So if you have a grow light for plants that don't get a lot of sunshine in a particular window or whatever, you'll notice that the light isn't always white. It might be like weird red and blue, which is what mine looks like. It's actually like a vibe. And that totally makes sense because chlorophyll A absorbs violet, blue, and red. And so that's going to work the best for photosynthesis. And then green is the least effective. If you get like a green light for your plants, it's not going to work. You might as well not shine anything on them basically because it reflects the green. It's not absorbing anything of that. So either a white light or maybe one of those like weird grow lights. And so the action spectrum for photosynthesis is a profile of the relative effectiveness of different wavelengths and confirms the effectiveness of violet, blue, and red light. So there are 2 different spectrums. Like the absorbance spectrum is just plotting the absorbance for different wavelengths. The action spectrum is plotting like a measure of photosynthesis with the different wavelengths. And so if we do this, it really does confirm like, oh yeah, if I shine the red or violet blue light, that's going to have the most photosynthesis. It's not just like a hypothesis. This has been shown. And so the action spectrum for photosynthesis is actually broader than the absorption spectrum for chlorophyll. Because again, like it's not just one kind of chlorophyll. There's chlorophyll A, there's chlorophyll B, and then there's other ones too. So the accessory pigments like chlorophyll B broaden that spectrum used for photosynthesis. And that slight difference, even though they're both like a chlorophyll A and chlorophyll B, there's tiny differences in the molecule structural difference that make it able to absorb, slightly different stuff. And actually, in the last decade, I love science so much because it's so interesting that there's still stuff we're learning, right? Like when you see stuff in the slides, like, oh, it is thought that, or oh, we recently learned that. Like, it's really true. So in the last decade, 2 other forms of chlorophyll were discovered, chlorophyll D and chlorophyll F, and they absorb higher wavelengths of light. And so, for example, in some organisms, they might be adapted to use these other pigments. So the cyanobacterium Crucoccydiopsis thermalis. I'm also being real about that. I don't know how to pronounce that. Crucoccydiopsis thermalis uses chlorophyll F instead of chlorophyll A in shaded conditions, which is genius. Yeah, it's just amazing. So there are also other accessory pigments called carotenoids. And you might think that sounds familiar. That is in carrots. So carotenoids appear yellow or orange because they absorb violet and blue to green light. So, yeah, carotenoids, carrots look like that, and they are orange. They look orange because of that. This is what they reflect or transmit, and this is what they absorb, violet and blue to green light. So that also helps broaden the spectrum for photosynthesis for plants that have carotenoids. Some are also photoprotective, so they absorb excessive light that would otherwise damage chlorophyll or react with oxygen. So now this all comes back to the light reactions. We took a tangent to learn about light, but it's for a reason. So when a pigment molecule absorbs light, one of its electrons goes from a ground state to an excited state, which is unstable. And so this is what this picture is showing. So if this is a chlorophyll molecule, and this is also kind of like a graph. So as a photon hits, it excites one of its electrons and it goes like to high energy. And then as it comes back down, in isolation, if this chlorophyll molecule wasn't like attached to anything, in isolation, the excited electrons will just fall back to the ground state, releasing excess energy as either heat or light. So if we see, fluorescence, that's like light energy that's coming back down from being in an excited state. So that's if you just had like a test tube of chlorophyll, yeah, on its own, not in its native environment in a thylakoid. So inside of thylakoids, there are what are called photosystems. So a photosystem consists of a reaction center complex surrounded by light harvesting complexes. The reaction center complex is an association of proteins holding a special pair of chlorophyll A molecules and a primary electron acceptor. So hopefully this will make sense in the next coming pictures. So a photosystem, like that. And then there's a reaction center complex. And then outside there are light harvesting complexes. And that reaction center complex has proteins with a special pair of chlorophyll A molecules and a electron acceptor. So let me just show you this picture. Okay. The colors are giving Barney. So this is the reaction center complex. And then the purple things are the light harvesting complexes. And there's more than one surrounding that reaction center complex. And the light harvesting complex has the chlorophylls. So that light harvesting complex has various pigment molecules bound to proteins, the purple proteins. And so they transfer the energy of photons. They like bop around all the energy of the photons. And then they transfer the energy to the special chlorophyll A molecules in the middle in the reaction center complex. So these chlorophyll A molecules are special, the ones in the middle, because they can transfer an excited electron to a totally different molecule. So these guys are like special. So a photon hits this pigment. It bounces around, and then it bounces the energy to this special pair of chlorophyll A molecules, and that can transfer an electron to something else, the primary electron acceptor. So this is how, we show it, but the actual structure is a lot more complex. But again, it's just easier to kind of show them as blobs. So this is what it looks like more. So a primary electron acceptor in the reaction center accepts excited electrons and is reduced as a result. Remember, like if something is reduced, it takes on extra electrons. So this solar-powered transfer of an electron from a chlorophyll A molecule to the primary electron acceptor is the first step of the light reactions. And there are actually two types of photosystems in the thylakoid membrane. They're numbered in order of their discovery. So they're not ordered in like how chronologically they work in the pathway, basically. So photosystem 1 was discovered first, and then they called it probably just photosystem. And then they saw the second one and then called that one photosystem 2. But in order in how they work, photosystem 2 comes first. Science is so funny sometimes. So photosystem 2 comes first, and it's called P680 because its reaction center chlorophyll A is best at absorbing light with a wavelength of 680 nanometers. Photosystem 1 is called P700 because its reaction center chlorophyll A is best at absorbing light with a wavelength of 700 nanometers. So that kind of makes sense. P for photosystem, and then whatever is the best absorbing light wavelength. Okay, and so during the light reactions, there are two possible routes for electron flow. There's the linear electron flow and then cyclic electron flow. And so we'll talk about the linear electron flow first. So linear electron flow is the primary pathway, and it involves both photosystems, 2 and 1, and produces ATP and NADPH using that light energy. So there are 8 steps in linear electron flow. Let me show you a picture really quick so that maybe it can help as I describe each of these steps. So this is a little bit more simplified. So this is photosystem 2. This complex, and then photosystem 1. So hopefully this helps, and then maybe, when you're doing your notes, you'll have to refer back. So step 1. A photon hits a pigment in the light harvesting complex of photosystem 2, and its energy is passed among the pigments. It's like playing hot potato. So step 2, an excited electron from P680 is transferred to the primary electron acceptor, and then we call this the oxidized form as P680 plus. So this photosystem complex is oxidized, it's excited, and so it's P680 plus. Step 3, an enzyme catalyzes the split of water into 2 electrons, 2 hydrogen ions, and an oxygen atom. The electrons are transferred to the P680 plus pair, reducing it back to 680. The hydrogen ions are released into the thylakoid space. An oxygen atom combines with another oxygen atom made by the splitting of a different water, And then together, those two oxygen atoms form oxygen, atmospheric oxygen, O2. And so that is how, you have to water your plants because it has to drive this path. And then your plants happen to form oxygen. And then that's why they recommend, having plants in your house, because it kind of helps provide more oxygen. Okay, so that's step 3. Step 4, those electrons that came from water are passed in a series of redox reactions from the primary electron acceptor of PS2 down an electron transport chain to, I think this is a typo again, PS1. So there is an electron transport chain here as well, and it functions in photosynthesis. So electrons are passed in a series of redox reactions from the primary electron acceptor of PS2 down a electron transport chain to PS1. That electron transport chain here includes this electron carrier called plastoquinone, or just PQ. Also to a cytochrome complex and a protein called plastocyanate, PC. The energy that's released by this electron transfer is used to pump protons into the thylakoid space, creating a proton gradient across the thylakoid membrane. So it sounds similar to something else, right? The potential energy that's stored in the proton gradient drives production of ATP by chemiosmosis. That also sounds familiar, right? Chemiosmosis. And so in PS1, like PS2, transferred light energy excites P700. So then P700 loses an electron to the primary electron acceptor there. And now we refer to it as P700 plus because it's missing an electron. And so P700 plus accepts an electron that gets passed down from PS2 via the electron transport chain. And then step 7, the electrons are passed from that primary electron acceptor of PS1 down a second electron transport chain to a protein called ferredoxin, or FD. Here, there's no proton gradient or ATP made by this electron transport chain. It's kind of just shuffling it along. And then lastly, the enzyme NADP plus reductase catalyzes the transfer of electrons from FD to NADP plus. And so 2 electrons are needed to reduce NADP plus to NADPH. The electrons of NADPH are at a higher energy level than they were when the electrons were in H2O, water. So the electrons in NADPH are more readily available for the reactions of the Calvin cycle. So that's why, it's not just water powering the Calvin cycle and using the electrons directly. They get shuffled along into this other molecule, and then they're higher energy, and then that can be used more easily in the Calvin cycle. And so this formation of NADPH also removes a proton from the stroma. So this is a figure showing all that. Photon hits this pigment, the energy bounces around, hits the special chlorophyll A molecules in P680. Water gets broken up, the molecules get put here and then transferred to the primary electron acceptor. Then they shuffle along to PQ, to a cytochrome complex. Here they make ATP in this electron transport chain. Electrons keep getting shuffled around, PC, then to P700. light is also hitting this photosystem, and so it's exciting this. The primary electron acceptor accepts this electron, passes it along to another electron transport chain. There is no ATP made here. It's just making, transferring stuff. And then you're making NADPH. So in that big picture overview of like the light reactions, this is how that happens. How ATP is getting made and then how NADPH is getting made. And they're both going to go into the next part, which is the Calvin cycle. So that was the linear electron flow. And so light reactions use solar power to generate ATP and NADPH. And that, those molecules provide the chemical energy and reducing power needed by the Calvin cycle to then make sugar. And the electron changes of the energy changes of electrons during this linear flow through the light reactions can actually be shown a little bit better in this mechanical analogy. So it's kind of trying to show you how it's transferring energy and electrons. Yeah. So first there's a worker here that uses a photon hammer. So here there's like a lever and there's an electron ball on this end. And then so with the energy from this photon, it like jumps this electron up 3 stories. And this is the photosystem 2 tower. So it's 3 platforms high, but that energy from the photon is what like yeets it up there. Another worker catches that electron at the top of the platform, puts it on like a mill conveyor belt that flows down the first platform to the next tower, the next photosystem. While it's rolling down that conveyor, this mill makes ATP, and ATP is released. So it's kind of using that energy to make ATP. The electron hits here in photosystem 1. Another worker uses a photon hammer and then yeets it up even more. And then there's a worker up here that catches the electron and then puts it into a bucket, making NADPH. So hopefully that helps. I think, yeah, it's not too bad when you think of like the big picture, okay, so this is how you're transferring one form of energy into like moving something else along. So that is linear electron flow. In cyclic electron flow, photoexcited electrons cycle back from FD to the cytochrome complex instead of being transferred to NADP+. So the electrons are passed to a P700 chlorophyll in the PS1 reaction center via the plastocyanin molecule PC. And so cyclic electron flow only uses photosystem 1 because it's kind of just like cycling it back there. It produces ATP, but no NADPH or oxygen results from this process. So this is a figure of the cyclic electron flow. So the electron goes from here to the cytochrome complex to here and then back here. And so several groups of photosynthetic bacteria have only a single photosystem related to either PS2 or PS1. For these organisms, cyclic electron flow is the only way to make ATP during photosynthesis. So this isn't in all organisms. And so photosynthesis may have first evolved in the ancestors of these bacteria in a form similar to cyclic electron flow. So we think cyclic electron flow is probably in part an evolutionary leftover in organisms that have both photosystems. Cyclic electron flow may have some photoprotective capability because plants that don't have it grow well in low light but can't grow well in intense light. So again, we kind of say like, well, we think it's probably this because we're still learning. And so if you notice some similarities between cellular respiration or parts of cellular respiration and parts of photosynthesis, it makes sense. So there are, similarities and then also differences, of course. So chloroplasts and mitochondria both make ATP by chemiosmosis. In the electron transport chain, in either one, there are electron transport chains that pump protons across a membrane as electrons are passed through carriers with progressively higher electron affinity. And ATP synthase couples the diffusion of the protons down their gradient to the phosphorylation of ADP to ATP. So that is what's in common in both chloroplasts and mitochondria. There's electron transport chains, proton gradients that are used to help make ATP. Some of the electron carriers, including iron-containing proteins called cytochromes, that also was similar, right? They are very similar in mitochondria and chloroplasts. The ATP synthase complexes are also very similar. But they're also very different in different ways. So photophosphorylation differs from oxidative phosphorylation in a few key ways. So in chloroplasts, the high energy electrons drop down the transport chain from water. But in mitochondria, they're extracted, those electrons are extracted from organic molecules. So it's kind of not like happening on its own in the way that it is in chloroplasts. And then in mitochondria, they transfer chemical energy from food to ATP. And chloroplasts transfer light energy into the chemical energy of ATP. So mitochondria is just like transferring chemical energy, but chloroplasts are making like chemical energy from something totally different, from light energy. And so although the spatial organization of chemiosmosis differs slightly, there's also some similarities in that. So in mitochondria, protons are pumped to the intermembrane space and drive ATP synthesis as they come back into the mitochondrial matrix. And in chloroplasts, protons are pumped into the thylakoid space and drive ATP synthesis on the stroma side of the membrane as they diffuse back into the stroma. So this is a figure kind of showing these concepts. Like some of the stuff that is similar, there is a membrane that's separating different spaces, and there is pumping of a proton by an electron transport chain. The proton builds up of pure, so there's a high concentration gradient, and so once the protons go down their concentration gradient, it powers ATP synthase to make ATP. So even though they are similar, they like are in different places, obviously, in mitochondria and chloroplasts, but this concept is really similar and interesting. So both ATP and NADPH are made on the stroma side of the thylakoid membrane, making them available for sugar synthesis in the Calvin cycle. So they're, what it's trying to say is that the way that it's organized in these thylakoids and why they're like flat pancakes is like what enables them to make the ATP and the NADPH be on the stroma side and then ready to just feed into the Calvin cycle. it's not like putting them in here and then you have to transport it out. It's making it so that the NADPH is on the stroma side and the ATP is on the stroma side and then just ready to feed into the Calvin cycle. Okay, so that was the light reactions. So part 1 of photosynthesis. Next will be the Calvin cycle.